Thank you very much to the organizers. It is absolutely fantastic to be able to present our work here. What I'm going to tell you a little bit about is the work that we've been lately pursuing with Dr. Bjornsson, where we are trying to gain some insights into the molecular basis of Kabuki syndrome via comparisons with related disorders. All the data that I'm going to show were recently published in this paper about a month ago. And so the starting point for our investigation was this observation that you've also heard from Dr. Bjornsson, which is that even though different Mendelian disorders of the epigenetic machinery are caused by variants in different genes, there are many shared clinical symptoms between them. And obviously, the clearest example of such a shared symptom is intellectual disability, which is shown in blue here and spans the entire wheel of disorders, as you can see. And there's also other such examples, including growth abnormalities, immune defects, seizures, etc. And so we have been wondering for some time now what may be responsible for these shared symptoms. The hypothesis that we came up with is that these shared clinical symptoms downstream of the distinct genetic defects may arise because there are shared underlying molecular abnormalities. And when I say molecular abnormalities here, what I mean is either disruption of normal chromatin accessibility patterns at specific locations throughout the DNA, or disrupted expression of specific genes. And just to give a more concrete, albeit still hypothetical example, if we suppose that in Kabuki syndrome type 1 or KS1, we have identified 3 genes that show disrupted expression, gene A, gene B, and gene C, and in Kabuki type 2, we have found 3 genes also with disrupted expression, gene C, gene D, and gene E. Then we would immediately identify this gene here, gene C, as being shared between the 2 types of Kabuki. And this would make it a strong candidate to potentially explain the clinical overlap between the 2 conditions. So this is basically the way that we were thinking about this. And so we devised an experimental approach that would allow us to test this hypothesis and detect such shared abnormalities. So here again, I am illustrating the idea for KS1 versus KS2. So we use mouse models for these conditions, and we compare the mouse models for Kabuki type 1 and Kabuki type 2 to their corresponding wild type mice. And what these comparisons allow us to do is to derive a catalog of abnormalities, which are found of molecular abnormalities found in the mice for Kabuki type 1 and the mice for Kabuki type 2. And then once we have done this for the individual disorders, we can then perform a statistical overlap analysis and derive a catalog of shared abnormalities. And then if this overlap hypothesis, which was our starting point, is correct, then we expect that these shared abnormalities will be particularly important for explaining the clinical symptoms that we observe in these disorders, especially those that are shared among them. So this is the conceptual foundation underlying our work. And so what we first set out to apply this approach by comparing Kabuki type 1 with Kabuki type 2 and Rubinstein-Taybi type 1. And it was known that patients with Kabuki type 1 as well as Rubinstein-Taybi exhibit hypogammaglobulinemia, in particular IgA deficiency, and also a reduction in mature B cell numbers. For Kabuki type 2, we didn't have such a good idea of what to expect because the immune defect was not characterized that well in the patients, but I'm going to return to that in a little bit. And we performed 2 molecular assays depicted here. Attack sequencing, which measures chromatin accessibility throughout the entire DNA, and RNA sequencing, which measures gene expression for every gene encoded in the DNA. I'm now going to walk you through some of our results. This here is a little bit complicated, but the x-axis, which you see ranges from 0 to 1, reflects the evidence that we have that a given promoter has disrupted accessibility in KS2. KS2. Promoters are locations in the DNA which are very important for controlling the expression of genes. And so a disruption in the accessibility of a promoter, we expect will have strong consequences. And so what we can see here is that if we focus on the red curve, which corresponds to those promoters with disrupted accessibility in KS1, then these have values concentrated close to zero, as evidenced by the height of the red curve around zero. What this means is that these promoters that have disrupted accessibility in Kabuki type 1 also have disrupted accessibility in KS2. About not every single one of them, but about 70% of them do. Whereas if we now focus on the blue curve, which corresponds to promoters whose accessibility is not disrupted in KS1, then we observe what is known as a uniform distribution, which means that these promoters tend to not be disrupted in KS2 either. Less than 5% of them are. This same picture is reinforced when we now look at the magnitudes of accessibility change in Kabuki type 1 and Kabuki type 2. Here, each dot corresponds to a promoter, and then the x-axis is the magnitude of change in KS1, whereas the y-axis is the magnitude of change in KS2. And we can see that when we have a positive change in KS1, we have a positive change in KS2 as well, and vice versa. What this means is that there is a broad and extensive concordance between the accessibility changes that we see in promoters in KS1 and KS2. And now, if we take those promoters whose accessibility is disrupted in both types of Kabuki, and we ask what happens in Rubinstein-Tabby, we observe the same picture. They, about 65% of them are also disrupted in Rubinstein-Tabby type 1. And again, the changes are largely concordant. As I said, these promoters are important for regulating the expression of genes. And so what we did next is to ask if this disruption in promoter accessibility is associated with a disruption in gene expression. So in all three of these plots here, the x-axis corresponds to the change in promoter accessibility, whereas the y-axis corresponds to the change in gene expression. And each dot corresponds to a promoter with its associated gene. And you can see again that there is a strong positive correlation. So in other words, meaning that when we have an increase in promoter accessibility, this is associated with an increase in gene expression, whereas a decrease in promoter accessibility is associated with a decrease in gene expression, which in other words means that these extensive promoter accessibility changes we are seeing have functional consequences as measured by gene expression. And this is just showing an example of a specific gene, which we thought was interesting. It's part 3B, which is known to play a role in immune responses, in particular to antigen presentation and recognition. So this also allowed us to perform a test of our starting hypothesis, which was that it is these overlapping molecular abnormalities that are particularly important for explaining the clinical symptoms. So what we did now is we separately looked at genes whose promoters show disrupted accessibility across all three conditions, KS1, KS2, and Rubinstein-Tabby type 1. And we compared them to genes whose promoters are disrupted, but only in one out of the three disorders. And if our hypothesis is correct, we would anticipate that it is the genes with a shared, whose promoters are disrupted across all the disorders that would be most likely to show a disruption in gene expression. And indeed, this is what happens. So here the y-axis is the percentage of differentially expressed or disrupted genes. The red dots correspond to genes whose promoters are disrupted across all disorders. The black dots are genes whose promoters are only disrupted in one of the disorders. And you can see that invariably across the three conditions, the red dots are higher than the black dots, which means that indeed it is these shared changes that are more likely to have consequences, supporting our hypothesis. However, what we were a little bit puzzled about is that we were not observing just a small number of big changes, but rather we were observing a very large number of changes distributed throughout almost the entire DNA, but each of these changes was individually subtle. And so what we wanted to see is whether it is this type of collective dysregulation, as we were calling it, that produces the clinical symptoms. And so we focused on the two symptoms of interest here, the abnormal B cell maturation and the abnormal IgA production. For B cells, we looked at genes that code for transcription factors, which are very well known to control and coordinate the maturation of cell types in the body. And we devised a metric which would reflect whether these transcription factors exhibit a dysregulation in their entirety, even though when we look at each single one of them, we may not observe a detectable change. And indeed, this is what we saw across all three disorders. So here, the red vertical line reflects the evidence that we have for collective dysregulation. And the smaller the value for the red line, the stronger the evidence. And you can see that it is always the case that the red lines falls outside the blue distribution, which is what we would expect by chance. And then when we moved to IgA deficiency, and this time we focused on all genes which are individually known to contribute to IgA production, then we again observed this collective dysregulation, but this time only for Kabuki type 1 and Rubinstein-Taybi type 1. When we looked at KS2, there was no evidence whatsoever. And as you can imagine, this was surprising given the strong clinical overlap that otherwise exists between the two types of Kabuki. And so it prompted us to measure IgA in the blood of the mice. These blood measurements actually confirmed what we were finding with our statistical analysis, which is that the Kabuki type 2 mouse model, which is these mice are shown here by the green dots, they do not exhibit an IgA deficiency. Their levels, the IgA levels, are comparable to what is seen in the wild type mice here with the black dots. Whereas when we looked at the Kabuki type 1 mouse model, we observed the IgA deficiency as has been previously described. In summary, what we are finding is that there is a large number of shared molecular alterations, both at the chromatin accessibility level, but also at the gene expression level between Kabuki syndrome type 1, Kabuki syndrome type 2, and Rubinstein-Taybi syndrome type 1. Now, of course, we have only looked, what I've shown is just data on these B cells, which are a specialized type of immune cell, but there are ongoing efforts in our lab to characterize this in other cell types and organs of the body as well. However, the clinical one implication from our data is that the clinical symptoms that we are interested in understanding arise not because of a small number of big epigenetic or expression changes, but rather it is probably a large number of small changes which together combine and produce these symptoms. In other words, it's what we would call a systems level dysregulation. However, it is important to emphasize that this does not mean that for an effective therapeutic intervention, one would have to go in and correct each and every one of these small changes. It is, in fact, I think, although we still do not have concrete evidence for that, just by interfering with a small number of strategically chosen targets is going to be enough. And in fact, there is precedence for this type of approach. And I'm going to mention here something which is very unrelated to Kabuki syndrome and has to do with predisposition to heart attacks. And while it is known that there is a very large number of changes in the genome which predispose one individual to an early heart attack, it has also been shown quite convincingly that just that giving a drug that just interferes with a very small number of these changes, and in particular controls LDL cholesterol levels in the blood, is very effective in preventing heart attacks. So I think a similar paradigm may be applicable to Kabuki and the other Mendelian disorders of the epigenetic machinery. And finally, as I said, we are in the process of performing this same cross-disorder analysis for other cell types and organs in the body. And we are also looking to do this kind of analysis for more disorders. And we think that this may actually help to narrow down the list of shared alterations. So I would like to thank Dr. Teresa Laperchio, with whom we worked very closely on this project, as well as Dr. Bjornsson and Dr. Kasper Hansen, who supervised this. Thank you very much for your attention. It is fantastic to participate in this conference, and I'm looking forward to answering any questions.